on applications of our microfluidic platforms serving as a sample prep units, really. And you know, some of the technologies, and I'll talk about two in particular. Microfluidic device that is designed for isolation of rare cells, can be really designed for isolation, immunoaffinity isolation of any type of cell. Those do not have to be necessarily a rare event. So this is one technology, and another one is isolation of extracellular vesicles. Sometimes you may hear people use word exosomes. And these two type of devices, they matured enough that we were able to create with the aid and support of KUMC and Kansas Institute of Precision Medicine. This is a COBRA type of award, what we call biomedical engineering core or liquid biopsy core, I'll tell you about. And we do process now samples in the core for PIs. We isolate those biologicals for them, we release them from the chip, and we send them back. And they are performing the validation of those biomarkers using their own, processing and, as I mentioned, well-developed, molecular biology method, sequencing, some type of molecular profiling, protein analysis. So it's going to be a great day today because you will hear from me as on those microfluidics as serving purely sample, a role of a sample preparation. But after the break, Katie Childers from Center will tell you about actually building integrated system where that sample preparation chip is just front runner of the whole integrated device. So I'm excited about the plan we prepared for you. So when you think about the biomarkers of interest, right, there are the whole number of those, there's a PSA testing, prostate specific antigen. You can have different, proteins tested, peptides tested. You can evaluate number of, the biomarkers at glucose level for what I care. Predicting the risk. They can tell us about the prognosis, right? They can tell us how to treat, whether there's, the liver needs to be increased or antistatins need to be increased. So if you think about conventional the way we analyze blood sample, right? There's no doubt in our mind that those are highly valuable, that we get highly valuable information based on analysis of those biomarkers. But today I wanna focus on two, which I call new generation biomarkers, and those could be exosomes or extracellular vesicles, and for the sake of the discussion, I'm gonna call them circulating tumor cells. And
when cancer spreads, tumor cells detach from the primary tumor and enter the bloodstream and travel to different parts of the body, a process known as metastasis. For patients with advanced cancer, presence of CTCs in the bloodstream is associated with prognosis. These circulating tumor cells are rare and until now have been technically very difficult to detect.
So this is a nice kind of illustration of what CTCs are. Not only these cells can be released from primary tumors, but also metastatic sites also release those. There's a fairly complicated process of changing the phenotype of the cell from, purely epithelial to mesenchymal. But there are quite a few technologies now that learn how to actually isolate those cells, because there is a value in learning how many of those events we detect in we can find there are in the blood. So one of the very early systems called cell search, that's by far, this is not actually now the only one that's FDA approved, but it was the first one. Here you see the probability of the survival curve for patients who were diagnosed with metastatic colorectal cancer. And you can see that, and this is the time from baseline blood draw, and the green line indicates that this is the probability of the survival for patients who in whom less than 3 cells, circulating cells, were detected per 7 milliliters of blood. So their overall survival is better than for patients for whom more than or equal 3 circulating tumor cells were detected. So this was one of the very first demonstrations and approved for patients diagnosed with metastatic breast cancer, metastatic prostate cancer, and colorectal cancer. For other type of malignancies, for example, pancreatic cancer, the sensitivity of assay was not as good to grant the FDA approval. But there are others. For example, nowadays, the new test detects those circulating tumor cells to assess the HER2 status. We know that, nowadays, it's not a breast cancer. There are different types, subtypes, and every one of those is treated differently, okay? So it's extremely important to learn about the, estrogen status, progesterone status of those circulating tumor cells, including HER2, because the treatment is different. And this is the whole idea of, I had in my title, precision medicine. That's the whole, the importance of detecting those biomarkers and learning about the characteristic of one's decision. One size does not fit all in those type of in treatment and maintenance of the treatment. Or, for example, there is an assay that looks at the circulating tumor cells and evaluates the androgen receptor in patients diagnosed with prostate cancer, because there is a clear indication that which type of therapy, whether there's even a point of providing the patients with the estrogen receptor targeted therapy if these are not present. So those are very valuable information that tell physicians how to treat patient, how to design and provide treatment that benefits best a patient. And circulating tumor cells can be one of those biomarkers that tell us about the nature of the characteristic of the disease. So there are a lot of modalities for the isolation of those cells. Electrical based based on the electrophoretic. And if you want to talk about this, you talk to Alex today about how their company, I forgot your name, but there's also a representative from the company, how they isolate those circulating rare cells, right? We can do, people do filtration. It's a fast method, but it's it's messy and dirty, and you isolate basically everything. It's fast, but does not give you the the benefit of getting really nice and pure population of the cells of interest. A big interest arrived, sparked by cell search on affinity isolation of those, positive affinity isolation of those rare cells. And those were initially performed in nanoparticles that were decorated with the antibodies introduced in the blood. And then based on those interactions, you use magnet and you target the nanoparticles. And since now cell is attached to this nanoparticle, right? You can isolate this. The problem is with the nanoparticles, there are a number of cells that actually love to engulf and eat those nanoparticles. So what you end up with is particles that truly attach to the surface antigen of a cell, but you're also gonna isolate the cells that will basically, for lack of a better word, literally they will eat the nanoparticles. So you get quite a low purity population of the cells. And then the whole in 2000, the microfluidic based assay for affinity isolation of those cells started to appear. I encourage you to read this review by my friend Matt Jackson, who composed a really very comprehensive a review paper on different methodologies for rare cell isolation. And those are, this is a figure from his paper, looking at affinity isolation basis, but different architecture of the device. And I could spend, honestly, 2 hours talking about the advantages and disadvantages of different type of architectures, but I'll just encourage you to take a look at this paper. But I'll tell you how we think that our approach, what benefits our approach provides, and why it stands out with respect to those other affinity-based isolation methods. So what do? We fabricate by injection molding, and this is we publish a number of the papers using this microfluidic technology with those what we call sinusoidal channels. They're very deep, 150 micrometers, and very wide, about 20 micrometers. What we call very high aspect ratio structures. What happens is after we bond, we perform covalent attachment of those antibodies on the targeting very specific subpopulations of the cancer cells that are present in the blood. And as the blood is infused, the architecture of those microfluidic channels enhances interactions of cells with the wall that's decorated with those antibodies. As you pass the blood, we just introduce raw, basically whole blood without any type of, preparation, fractionation, modification. Those cells interact with the wall, and they're retained by the interactions with the antibody. You can enumerate those cells, which will tell you, I just showed you, right? There could be a prognostic information, what's the progression-free survival or overall survival for the patient. But you can release those without staining and perform a whole slew of different molecular analysis. So what is the basic of this really methodology? You have those sinusoidal channels. It's like putting those cells on a centrifugal, basically influence. Those cells are influenced by centrifugal forces that are induced as the liquid passes and makes those frequent right or left turns. So larger cells are more efficiently brought to the surface of the microfluidic channel, and as they as they see the channel wall, we have what's called effective roll. They roll for at least 500 micrometers. So the probability of actually meeting that antigen on the surface of a cell and interacting with the antibody that's attached to the surface is quite high, but it's also dependent on how fast we flow the blood. So you can see that for very fast processing, 4 millimeters per second, that application, that's about 150 microliters per minute, you have about 70% probability based on the COMSOL modeling and simulations of the process. But if you decrease the processing to 0.1 millimeter per second, virtually you can capture every cell. The problem is that if I lower the processing, I suffer low throughput. And there's another consequence that when I go really slow, I'm exposing this assay to a lot of non-specific adsorption of cells, of any white blood cells. And there are, billions of those, millions of the white blood cells and billions of red blood cells in 1 milliliter of blood. So as a consequence, what we do is you actually process faster at 2 millimeters per second. You get about 90% recovery for a cell that's presented with about 50,000 antigen molecules on the surface. And by doing so, the shear rates of the blood with the surface of a microfluidic are high enough that it will strip any non-specific adsorbing cells, but they're not strong enough for any antigen-antibody bonding. You need to remember antigen antibody are the strongest non-covalent bonds established. There's a lot of interactions, hydrophilic, hydrophobic, van der Waals interactions. They will keep the antibody antigen attached while washing away all the non-specifically bound surface. That high shear stress when you process this non-Newtonian fluid as blood will give you the very high purity of isolated circulating tumor cells. I talked to you about this already. You can design different chemistries. When we started with direct attachment, you can strip those cells from the surface. We're using trypsin, but the problem is that trypsin will digest any markers, protein markers of the surface. So if you're interested in analyzing membrane proteins, for example, that is not a good way of doing this. So there are those different chemistry, whether the using enzymatic, based on enzyme that you can release the cells, or remember right now the sort of workhorse of ours is this photocleavable linker. So those are just an examples where, by changing that antibody that's attached to the surface of a microfluidic, you can target different type of cells, right? So I'm showing you, for example, anti-EpCAM antibody, you can target epithelial type of cells, but you can use anti-CD138 antibody and look for myeloma cancer cells. You can look at the mixed monolayer, what we call, you perform a monolayer of 3 different antibodies, and you could target, for example, acute myeloid leukemia cells, or process on 3 different chips and look for which population or subpopulation is the dominating one when I process the blood. So there are a lot of examples where we utilize this technology for the isolation of either rare events or not necessarily rare events, but that gives us fast processing time, gives the PI, pure population of the cells, that can be analyzed fairly quickly in downstream processing. Another biomarker of interest, which if you look probably now at the interest, beats the cell probably twofold, are extracellular vesicles. Those are very small particles that historically, if you read it, quite actually early, I think in 40s, there was 1940s, there was a paper that scientists observed those nanoparticles, about 100 nanometer particles that were released by cells. But the initial take was that those are the particles that basically are released by the cells, that they contain impurities of no value whatsoever. That dramatically changed in the last, I want to say, decade. Now we know that actually not only those nanoparticles, those exosomes or extracellular vesicles, they release when they're released, they contain the cargo that is specifically to the mother cell. Furthermore, those exosomes, EVs, they actually they act as a role of a postman. Actually, they communicate, they carry the information and the message contained in those particles to other cells. So the interest in analysis of those, the interest in enumeration of those, and finding which type of extracellular vesicles are actually disease specific is really vast. So we employed this again, immunoaffinity isolation or affinity-based isolation, which, so far I told you those are, I referred to antibodies, monoclonal, polyclonal antibodies, but there are other affinity agents. You can use very elegant aptamers, right? There are a whole number of advantages of using aptamers, much more stable at room temperature than antibodies, right? Much smaller, so you can pack more of those per unit area on the microfluidic surface. So there are a lot of advantages. There are peptides that are investigated. So there are other type of affinity agent that you can consider certainly in your assays. A lot of research in our in the center were directed using for affinity isolation using antibodies. So for the isolation of the extracellular vesicles, again, there's a lot of methods that are being reported. You have size exclusion chromatography. They work, but those columns are quite expensive actually. So if you think about the, clinical application, right, and the cost of the assay, you do have to, even though it may not be the priority, the first thing you think about, but that has to be in the back of your mind when you're designing those assays to make sure that they are affordable, that even if they are not covered by the insurance, right, individual can, the researcher or clinical lab, they can afford to employ this method to actually validate biomarkers. So the size exclusion chromatography, ultracentrifugation, but a new ones developed is basically precipitation with polyethylene glycol, where you just think of those EVs as little balls of protein, right? Just larger, just a ball of protein, and they're being precipitated as polyethylene glycol is added to the solution. Ultrafiltrations, certainly you can, but you tackle all the EVs. And remember, every cell in my body is releasing EVs. So I said something that's really important. Which type of those, which phenotype of the EVs are actually disease specific? So ultrafiltration, ultracentrifugation, size exclusion chromatography, precipitation, those are good methods, but they will isolate all the EVs in the sample. Again, immunoaffinity stands out because we have a way of actually looking at the very particular subpopulations of EVs. Remember, I said they come from mother cell, and the cargo is similar to the cell from which they were released. They will contain the protein antigens, those lollipops on the surface that are characteristic of the mother cell, and that's how we utilize those characteristics of the EVs. And here are just a few examples where these applications used targeted tetraspanins. Those are proteins that are detected in EVs and exosomes. Those could be clusters of differentiation 81, CD81, CD63, or CD9. So you create monolayers with antibodies targeting those antigens, and you perform immunoaffinity. Again, this is great, but pretty much every exosomes or EV has CD81. So you're not going into much more differentiation. There's a nice paper with this very particular, very characteristic signature coming from Toner and Shannon Stott group at Harvard. This is called this EV herringbone chip that by introducing those what's called herringbone structures, they introduce in the during processing sort of microvertices. It helps with mixing. If you paid attention when Dr. Supper was introducing this, doing his introduction, he said that, you know, mixing in microfluidics is hard. Reynolds numbers, right? The number that tells you whether you have laminar flow or turbulent flow are less than 1. They're not even less than 2,000. I can tell you they're less than 1. So there is no mixing unless you have time for this to just stay and diffuse by mixing. So this is an elegant approach with those herringbone structures to actually induce mixing of the fluid in the microfluidic. So so these devices were not only used for cells, but also demonstrations for extracellular vesicles. And you can read about the metrics. We certainly look at the high throughput. What is the volume of the sample that can be processed in a certain amount of time, right? Faster is better, of course, because within a 8-hour workday at a clinical lab, you can process more samples, right? So that's a good metric to pay attention to. What is the sample volume that you can process? Are you stuck with few microliters only, or actually you can extend your assay to 10 milliliters, for example? So those are the metrics that are absolutely essential when you evaluate the assay, and you find a way to demonstrate the strength of this technology. So this is a different now architecture of a device. Now you're looking at, remember, for cells, we had those sinusoidal channels. For the extracellular vesicles, now the architecture contains to be literal, this chip contains actually close to 1.5 million posts, or pillars as we call them. They're very densely populated. This this concept came actually very early when I started working in Soper's lab, and we started optimizing solid phase extraction of nucleic acid. By introducing those pillars, so those posts, what you're doing, you're increasing the internal surface area. So the footprint of a chip does not change, right? But the surface area does. It gives me a lot of benefits. One of them is, I have quite a large dynamic range. So what does that mean? That if I process 1 milliliter of the sample, I am not saturating the chip with the biomolecules of interest. I can process a large it's just pure analytical chemistry, right? I can process large sample and I'm still going to effectively and efficiently recover the biomolecules, right? The footprint does not change. Just the surface area of this ship is 38 centimeters square. And I always say that this is SI metrics. This is 6 square inches. I'm gonna say it. That's 6 square inches internal surface area. That's why you have a capacity to isolate. Just if you calculate based on size, 4 times 10 to the 11th particles in this chip, with a 1 inch by 1 inch geometric size. So those are the design considerations that you have to come up, think about. But, and again, the idea is we covalently attach antibodies to that chip, and we perform the processing of the plasma, fish out based on immunoaffinity, the EVs of interest. But designing this is not you need help with the design. And again, we talked about, there's this one slide in the introduction that very often you have to start with some kind of simulation. We do the COMSOL based analysis of okay, what is the spacing that I need that will be necessary between those posts to secure efficient isolation? What is the size of those posts? Right? That gives me some kind of density. If I have that type of size and spacing, what would be high throughput analysis? If you make it super dense and as a consequence, your high throughput will be, I don't know, I'm going to exaggerate, 2 microliters of a sample per hour. This is not really reliable, right? And analysis that can lead to vast clinical applications and sample processing in a high throughput format. So this is really important to use those tools, learn how to use those tools. So I'm not gonna go in vast details, but that's how we started designing this microfluidic for extracellular vesicle isolation. You look at okay, what do I need to consider? Okay, I know we're gonna process sample by hydrodynamic flow. I know that. I need to think about the, convective transport of the exosomes or EVs, right? I need to think about the diffusion of those. What is the rate of the diffusion of those big balls of protein, really, right? What will be the interactions of the antibody and antigen? Like how strongly do they bind, right? Is the Ka or Kd a law? Is it in nanomolar range, or is it 1 micromolar range? That makes a big difference, right? Then you create what's called a simulation, right? In either Monte Carlo model, as we use is here. And you have all those metrics and you design, okay, if I'm going to introduce in a certain size microfluidic channel, you can treat the spacing between those posts and microfluidic channel, right? So what is the probability that if I put 10 of those EV molecules, what is the likelihood that they will actually meet the wall and interact with antibodies rather than just pass through? You can imagine that if you employ very fast processing, very fast linear flow, right, millimeters per second, they will just pass the microfluidic without even opportunity to meet the wall. They need time to diffuse to actually the surface. So this is the balance between how to let those EVs diffuse and have a probability, have a chance to actually meet the antibody, and processing rate. So I don't spend 1 workday on one sample. So those are the criteria we look, right? How does the recovery, what's the efficiency, how is the efficiency changing when I'm changing those parameters? How fast I'm saturating the surface. So you can basically model those processes, and you have here's the figure that kind of represent this. You see this is the pillar. You treat this as a channel. You introduce the each one of those traces represents a path of one extracellular vesicles. When it's marked with a cross, that means it met the wall and it's a likelihood it met the antibody. When it's 0, it passed the length of the channel without being without interacting. So you can process and model the recovery based on the data. You can see that you need to process sample at 6 microliters per minute to get about 95% recovery of the cells. If you want to compromise and you're willing to compromise, you can increase that processing rate to 13, and you still can isolate 8 out of 10 EV particles. So that's how you design, and that's how you come up with the architecture that eventually consists of 1.5 million pillars with 10 micrometer spacing and 10 micrometer it's not really diameter. Those are the square shape, but basically side of the pillar. So what do you do with this, right? Well, you have to evaluate. Remember, we talked about the specificity of the isolation. Never ever rely on only one method to evaluate that process, that metric. We used nanoparticle tracking analysis to evaluate, okay, how many particles we isolate when we have device modified with the antibody or its isotype. Isotype is the same class of antibodies, but targeted, for example, for animals. So these isotypes we should never isolate the human type of, for example, exosomes, right? So you compare antibody and isotype using different methods. As I said, nanoparticle analysis, we looked at what is the concentration of the RNA when we isolate those and we purify the RNA from those particles. And then we looked at further, okay, we have RNA. Are there specific differences? Are there significant differences between when we perform gene expression analysis of the different transcripts? So you can see here that when we look at the gene expression and this white panel, this very bright, very pale panel here is the basically abundance of specific genes when we isolate some type of particle on using isotype versus chip that was modified with either anti-EpCAM, anti-FAP, or anti-CD81. You can clearly see the differences. The pale yellow means there are very few or none of the transcripts of mRNA enclosed in those EVs. The bright red tells you that there are quite a lot of those transcripts, right, per nanogram of the total RNA. So that's how you analyze and you determine specificity. You look at you want to visualize, right? You use transmission electron microscopy to actually visualize the particles that are isolated and released. So you can see here particles that are released from anti-CD81 bed. This is just isotype, nothing there. This is the negative control. Do not rely only and exclusively on one method, because if I would look at the NTA, I would not maybe have courage to go into the molecular profiling, because maybe, just maybe, the NTA does not give you enough, a discrimination about what type of particles we have. So how can you utilize those EVs? And I'm gonna just give you one example. We isolated extracellular vesicles from plasma of patient diagnosed with breast cancer. There's a hospital-given test, it's called Prosigna, that can distinguish based on tumor tissue collected from the patient what type of subtype of the breast cancer there is. The problem is, for not always, this is the project in collaboration with KUMC. Women travel even when sick. Sometimes they initiate treatment in facilities and hospitals that are actually not equipped with having a tissue bank where the primary tumor tissue can be actually evaluated for those type of tests. So again, because the EVs are released from particular type of cells, and we know now what specific antigen to target, we decided to evaluate the molecular cargo of EVs diagnosed with patients with different type of breast cancer. And we did. So you're looking here at the molecular profiling using that Prosigna assay that we employed for the EV total RNA. And you can see dramatic differences. Again, red is highly abundant transcript. Pale yellow is pretty much none of it. But based on this type of heat map, you can classify those samples into different cohorts. And what does that give you? For each different subtype of a breast cancer, there is a different treatment. Some patients do not need chemotherapy. Maybe, hormone therapy will be enough to fight the cancer. But there are other type of, for example, HER2 enhanced, that definitely need very strong and very aggressive type of treatment. So if you can classify, if you can tell about the disease based on simple blood draw and then processing of a sample, a plasma sample, isolate EVs, analyze, and navigate patient to a very particular, very specific type of treatment, it's a powerful idea. So this is just an example of that. The great thing about the design of those microfluidics, and we owe that to sort of collaboration and a want of a commercial partner who wanted actually to commercialize those devices. So Biofluidic designed the operation of those microfluidic with the aid of a robotic system. So we kind of remove the operator out of the equation. We remove, hydrodynamic pump, syringe pump out of the equation. We can put 8 samples at a time and process on the robotic system, set up a standard operating procedure for processing, wash, release, and send this to the clients. So that's a very, very strong transition. When you have a technology and then you design mode of operation, you can actually not only produce a lot of those devices, but you can analyze a lot of samples in a high throughput fashion. So this is, I'm going to conclude with this. This is the KU Medical Center. It's in Kansas City, Kansas. We're in Walhall East, one of the building there. We have our liquid biopsy core. Last year, the University of Kansas Cancer Center was recognized as a National Cancer Institute designated comprehensive center. So this is a big deal for us. With that, I'm going to conclude this talk, and I hope, I'm giving you encouragement that you can design those assays, and you're in a place where with the closeness of the cancer center, KU Medical Center, you can build collaborations with people, with physicians, and test those designs, test those microfluidics with not only cell lines, but also advance into processing clinical samples. Thank you so much. Are there any questions?